Hi, I'm Alastruey, and I'm delighted to talk about how we can use datasets of simulated pulse wave signals to conduct research on pulse wave analysis using machine learning models. I would like to thank Rio Tori and Nikos Stergiopoulos for inviting me to present in this session. I lead the hemodynamic modeling lab at King's College London, where we study blood flow in the cardiovascular system using clinical data and a combination of computational and experimental modeling. Our broad aim is to develop methods for the assessment of cardiovascular function based on the analysis of pulse wave signals, such as blood pressure and flow velocity. Pulse wave signals are produced by the propagation of the pulse wave, which is the result of the beating heart interacting with flexible arteries, such as the aorta. Using computational blood flow modeling, we can simulate pulse wave signals at common measurement sites in the body. But why do we want to simulate and study pulse wave signals? These signals can be measured in vivo using a variety of devices, including wearables, and are influenced by the heart, vasculature, and the respiratory and autonomic nervous systems. So that makes pulse wave signals a rich source of information to assess human health, including cardiovascular health. This is the outline of my talk. I will start by describing how we can create databases of simulated pulse wave, representative of samples of real subjects. And then I will show you some clinically relevant studies on how in silico pulse wave datasets can help us develop pulse wave analysis algorithms involving machine learning models. The development and testing of algorithms for analyzing pulse wave signals requires comprehensive datasets measured in large clinical cohorts. And unfortunately, acquiring these datasets is usually a complex task. On the left, you can see some of the issues that we may encounter. To facilitate this process, we have created datasets of simulated pulse wave representative of samples of real subjects using one-dimensional blood flow modeling. I will now briefly describe the methodology we use to create our dataset with over 4,000 subjects aged 25 to 75 years old under normal physiological conditions. It consists of 5 main steps. First, we simulated the pulse wave signals for a 25-year-old healthy subject that we refer to as our baseline subject. The cardiovascular properties for this subject already existed in the literature. Second, we reviewed the clinical literature to determine how model input parameters change with age. For each parameter, we had a mean value in solid lines and a standard deviation values in dashed lines indicating plus or minus 1 and 2 standard deviations. Third, we identified the parameters, one per row in the figure, that strongly influence pulse wave. By increasing in blue and decreasing in red, excuse me, each parameter independently by 1 standard deviation from its baseline value, and then assessing the corresponding changes in pulse wave morphology. For example, at the carotid artery on the left and the radial artery on the right. Fourth, pulse waves were simulated for a baseline subject at each age using age-specific mean values for all parameters found when reviewing the literature. As an example, the red circles indicate the mean values for the 45-year-old baseline subject. In the final step, the parameters previously identified as having a strong influence on pulse wave were varied at each age by plus and minus 1 standard deviation to obtain thousands of virtual subjects. Once the database was created, we verified that the simulated pulse wave signals are representative of samples of real subjects. And we did that by comparing simulated pulse wave with in vivo data from healthy subjects from the clinical literature. Here you can see comparisons in a few arterial locations, showing similar changes in pulse wave morphology with age to those measured in vivo. We also compared indices calculated from the simulated pulse wave across age groups with corresponding in vivo indices from populations of real subjects. And we found that the trends and even the values in most of the cases were similar between the two. For the remaining of my talk, I would like to show some clinically relevant studies on how in silico pulse wave datasets can help us develop machine learning-based pulse wave analysis algorithms. In silico pulse wave datasets are useful for designing and testing algorithms of pulse wave analysis, including machine learning models, understanding the potential shortcomings of existing technologies, and also understanding the mechanisms underlying hemodynamic observations. I would like to illustrate these points with 2 clinically relevant studies. The first study is the detection of abdominal aortic aneurysms, or AAAs, from pulse wave analysis. These aneurysms are usually asymptomatic until rupture, which is unfortunately associated with very high mortality. Therefore, early detection of AAAs is of paramount importance in reducing mortality. But the problem is that most AAAs are detected by medical imaging incidentally. So we're investigating post-wave analysis methods based on machine learning that are more convenient and also less expensive for large-scale screening than medical imaging exams. We have created a machine learning architecture based on the recurrent neural network to detect AAAs from PPG post-wave. Our method was trained and tested using the PPG signal measured at the finger from a data set of over 10,000 virtual subjects with and without AAAs. The model had a very high sensitivity and specificity in early detection of AAAs, as the confusion matrix and receiver operator characteristic curve show at the top. We then simulated a more realistic scenario during testing by adding random noise to the PPG pulse wave. That reduced the sensitivity and specificity to above 86%, which still suggests that the trained machine learning model was competent in classifying normal and aneurysm conditions using a digital PPG wave that had less resemblance to the pulse wave of the training set. The number of false positive and negative results increased with increasing age and decreasing aneurysm size, respectively, suggesting that these two factors should be considered to optimize the predictive power of machine learning algorithms. So the in silico dataset seems to be useful when it comes to the design and testing of machine learning models. The other clinically relevant study that I would like to show you is the estimation of the stiffness of the aorta, which is an important marker of vascular health. Indeed, increased arterial stiffness has been shown to be an independent predictor of morbidity and mortality from cardiovascular disease. And it is one of the biomarkers of vascular age. If you're interested in this topic, I suggest having a look at the cost-action vascular age network, which is very active in studying vascular aging. Aortic stiffness cannot be directly measured in vivo, but we can use the velocity at which the pulse wave propagates in the aorta as a surrogate for aortic stiffness. And several methods have been proposed to measure pulse wave velocity from pressure or flow waveforms measured at two arterial sites. For example, those shown in the sketch on the left. Typical combinations of waveforms are shown in the center, and the sketch on the right illustrates the foot-to-foot method for estimating aortic pulse wave velocity using two pulse waveforms, which calculates pulse wave velocity as the ratio of the distance between the two measurement sites to the time delay for the foot of the pulse wave to travel from one measurement site to the other. Using a database of over 3,000 virtual subjects, we could test existing foot-to-foot methods and show that only the carotid femoral pulse wave velocity is a good indicator of aortic stiffness, which is in agreement with clinical studies. We could also provide a mechanistic explanation of why this is the case. Results on the left show that carotid femoral pulse wave velocity correlates well with aortic pulse wave velocity across a wide range of values of true aortic stiffness. However, on the right, we can see that brachial ankle pulse wave velocity overestimates aortic pulse wave velocity, since it is related to the stiffness and also the geometry of more peripheral muscular arteries than the aorta. And carotid radial pulse wave velocity and femoral ankle pulse wave velocity do not capture aortic stiffening. They are therefore not so good surrogates for aortic stiffness, which is in agreement with some in vivo studies. The carotid femoral method is an accurate method that is widely used in clinical research. However, it requires skilled operators to acquire the data, and hence its applicability is limited to the clinic. Using a combination of in silico and in vivo data, we are investigating methods to estimate pulse wave velocity from a single pulse waveform. For example, the radial pressure waveform in the arm. We follow two approaches, either by using fiducial points extracted from the waveform as input to a Gaussian process regression algorithm, or using the whole waveform as input to a recurrent neural network with long short-term memory. Here you can see scatter plots of estimated versus measured pulse wave velocity, with the estimated value obtained using either Gaussian process regression on the left or a recurrent neural network on the right. And these are the corresponding Bland-Ullmann plots. The average root mean square errors for all the estimated pulse wave velocities for the over 3,000 in vivo subjects were smaller than 1.8 meters per second. The in silico pulse wave dataset allow us to examine the machine learning models with an exact theoretical reference value of the carotid femoral pulse wave velocity for each virtual subject. It also enabled us to investigate the effect on the estimated values of pulse wave velocity of adding noise to the radial pressure waveform in a controlled way. Without any noise, the average root mean square errors for all estimated pulse wave velocities using the recurrent neural network was smaller than 0.1 meter per second for the over 4,000 virtual subjects. This increased to 0.2 meters per second when 20% of random noise was added to the pressure waveform. These results show the possibility of assessing arterial stiffness using a single peripheral pulse wave instead of the two-point carotid femoral method. To conclude, our results suggest that vascular function can be assessed from a single pulse wave measure in the arm using machine learning-based pulse wave models. In silico pulse wave datasets provide accurate theoretical knowledge of the anatomical and hemodynamic properties being examined, as well as the full control of the experimental error present in the input pulse wave data. As a result, in silico pulse wave datasets provide an alternative way for the development and preclinical testing of pulse wave analysis algorithms, including machine learning-based algorithms across a wide range of cardiovascular conditions and in a relatively quick and inexpensive manner. And lastly, in silico testing enables us to understand physical mechanisms underlying correlations observed from populations of real subjects and train machine learning models across a wide range of physiological conditions. I would like to thank my group and funding bodies, which have all contributed to this research to some extent, and thank you for your attention.